got stopped. If you are performing some molecular docking, usually while in the starting of this video, you might have seen that you will be exploring the binding cavity, which is only exterior part, which is outside of the protein. Okay, but once you see the video, you will realize that the actual binding pocket is not present outside of the protein. The drug has to travel inside these protein loops, inside these alpha loops, alpha helices, and where this drug molecule will reach to the center of the protein, that is the actual binding pocket. That is when the drug is actually showing its biological activity. It will be done when you are actually doing some MD simulation, not the molecule docking. Can you see that? It took almost close to some extent of time where the drug has reached to the center of this particular protein, where the actual binding pocket is located somewhere here. If you do only the docking, where you will explore only this particular part, which is not the actual binding cavity of your target macromolecule. That means whatever MD molecular docking you have done, it is of no use until or unless if you explore that particular chemical substance and target protein in MD simulation, you are not actually getting the actual mechanism of action. To know that, you have to definitely perform MD simulation. Then what exactly happening in MD simulation? These are the typical steps in MD simulation. Basically, you'll be preparing the molecules, both ligand molecule and protein structure, for making the geometrics ready for MD simulation, and you are optimizing some force fields, and you'll be getting a minimum energy conformation. Then after that, to actually give the reality to the protein structure and ligand structure, it is just like you are doing in vitro assay, where in pharmacology you might have seen there are organ baths are there in pharmacology laboratories, where we are keeping thyroid solution or ringer solution to establish the same kind of biological environment like human being. Okay, even in physics also we'll be taking dissolution apparatus where we are maintaining 0.1 molar HCl or some kind of pH buffer to actually match with our biological system. Similar to that, in the simulation also, in the computer in silico modeling also, we will try to give the same kind of temperature like our biological system, and we will give even salt composition also, and the pH also we will maintain in the system computer. And later we'll be performing the dynamics depending on various predefined parameters. We are calling them as NVE, NPT, and NBT symbols. And later we are analyzing some data whatever obtained from there. So with that, we will actually get some useful information rather than just only getting interaction profile. Okay, I'll show you just a few quick videos to summarize this MD simulation. After that, I will show you hands-on. So this is just a system before running the MD simulation, and I just want to show you some screenshots. This was taken from Desmond software, where there are even water is also involved in the binding cavity when the drug molecule. This is basically synthetic material which is present in a binding cavity of some protein molecule. These are actually taken snapshots over a period of time, over a period of 100 nanoseconds time. You can see that in each and every stage, the drug actually changing its conformation. Not only that, you can see that this red color loop is changing its position. In all the 6 frames, you can see that this red color portion is actually dancing. And even the drug molecule is also actually getting a different orientation. So it is acquiring a different conformation where it may actually achieve the minimum energy conformation. If it is achieving minimum energy conformation, you can believe that that is the actual biological pose or biological conformation which is happening in our actual world, in our human body. So these are all interaction profiles. Let me show you one again video. Let me skip all these things. This is how interaction profiles are established. This is one video. Just I think so. It's a matter of 1 minute, but I'll quickly play through. Can you see that the protein is jumping and dancing, and where the ligand molecule is also dancing, and it is changing its posture. And similarly, I told you about this red loop. This is the N-terminal chain. Similarly, there will be C-terminal also. How it is changing its position. And again, similarly, there are green loops and red loops are yellow loops are there. And this loops, how it is making this particular chemical substance to change its conformation. So over a period of 100 nanoseconds, there will be chances where this particular compound will achieve a proper energy minimized pose, and that will be the actual binding pose which is in human body. Let me show you the interaction profile. This is what actually happening, where the water molecules are also coming into the binding cavity. You can see that always there is bonds are happening, bonds are forming, and the bonds are breaking. So it will happen where it will basically addresses this particular approach called as rate theory. According to rate theory, there is one concept that any drug molecule has to form n number of interactions for a fraction of time length. For example, if you take 1 second, for a period of 1 second, there might be definitely more than 1,000 times the bonds are forming and bonds are breaking. Still, that particular compound is present within the binding cavity. Means you can assume that this drug will try to stay some more time in the binding cavity to give the biological response. So it is not like only one time interaction happened and the biological activity will come. It's not like that. So this will solve actually this rate theory of drug action, which is an important concept. So these are actually various qualitative parameters to understand. Usually we're always looking for this red line to be a straight line. If it is a straight line, we will say that the protein complex is stable, protein ligand complex is stable. If it is stable, we can say that yes, this particular compound can be taken to the next stage of exploring in vitro or in vivo studies. Similarly, there will be even RMSF and RMSD pictures will be there. They will tell you about these fluctuations are minimal so that the protein ligand complex is stable. Similarly, interaction profiles are there, where in the docking process, you'll be getting only one frame, but in MD simulation, you'll be getting more than 2,000 frames, sometimes even 5,000 frames. In each frame, there are multiple interactions, and we can actually take that interaction fractions over 100 nanoseconds time scale. These interaction profiles will tell. If you ask me, among these three, this is the actual co-crystallized ligand, these two are the explored molecules, and I will say that this particular molecule is having n number of interactions compared to co-crystallized ligand, and this will be better inhibitor than co-crystallized ligand or a standard inhibitor. So these are actually softwares which are available for free to some extent, and some are actually paid versions for performing this molecular dynamic simulation. Amber, which is not free, NAMD, which is free software, Gromax is free, Desmond is free for academic people, and also you have Charm, which is not free, LAMPS, which is not free, VMD, which is free software, and all these softwares can be explored for running this MD simulation. I think so. With this, I'll stop the presentation and I will show you the demonstration. Before jumping into demonstration, if you have any questions, you can ask me.
Sir, I have actually two, three questions. May I ask, sir?
Yes, sir. Please go ahead, sir. If
Okay.
the organizers are permitting, because I thought of finishing the presentation within 1 hour and showing half an hour demonstration, but right now it is 12 p.m. exactly. If it is allowed, I will try to show the demo quickly, and also I'll take the questions now first. Sir, please, you can ask, sir.
Okay. Okay. Actually, you shown that network is built.
Yes,
and
sir.
networks is having different nodes and edges.
Yes, sir.
You told only hub gene and mid gene. You mid targets you taken. Why we are not taking peripheral targets? If we are want to select actually, that can be taken. That may also have a chance to.
Usually, for representation sake, the people are actually showing some genes on the peripheral region. It's
Yes.
not like that actually. The selection is based on three things. One is
Yes.
called as betweenness, closeness, and nearness. Depending on these
Okay.
three properties only, we will be selecting. And there is one more filtration criteria in network pharmacology that is called degree filter.
Degree filters.
Degree filter. Depending on these four parameters, we will be selecting the target molecules for exploring
Okay.
further either in silico, in vitro, or in vivo.
One is the betweenness, closeness,
Closeness, nearness, and the
nearness,
last one, degree filter.
degree filter. Okay.
Degree
So,
filter, actually,
okay.
according to my personal experience, degree filter will give you a lot of information
Okay.
because
This.
you need to
Okay.
sometimes address about the off-target effects. So in the
Okay.
network pharmacology, sometimes it is deceiving the people. Why I am telling
Mhm.
deceiving? Sometimes people will feel that okay, there is a protein molecule which is connected with more than 100 proteins, which is
Mhm.
connected with 100 different pathways. But if you're
Mhm.
targeting that particular protein, you're doing definitely a mistake if you're not reading or not analyzing that particular target properly, because that target, if you are inhibiting that particular target, which will definitely give you n number of side effects, because it is connected
Mhm.
with more than 100 different pathways, it definitely not an ideal choice of exploration.
So it should be edge should be less.
It should be
Edges.
less, But again, there is a criteria. Sometimes what researchers will do, it is again personal opinion. It's my personal
Yes.
opinion.
Yes, sir.
Sometimes I will try to apply the degree filter with minimum 10 connections and maximum of 20 connections. Even though there are some proteins, they have connections over and above 50 and 60. Even though it is a good valid molecule, valid protein structure, I will try to avoid exploring that protein structure because definitely there will be off-target effects. Myself, I am purely in silico person and I am not performing any in vitro in vivo assays. But even then, definitely people will ask you question. If your protein target is connected with 50 other proteins, then definitely there will be off-target effects, which may show you severe, the drug-drug interaction, not drug-drug interactions, and associated, the side effects will be definitely come.
Okay. Okay. So one more thing is that people are generally working on the human disease only.
Yes, sir.
We are just ignoring animal plant diseases. Okay. So this philosophy also we can use for that purpose. But challenge which I found actually, we
Yes,
are
sir.
not getting genes, we are not getting proteins, we are not getting 3D structure of that. So is there any solution you might know about the things how we can proceed actually?
One concept called One Health. You might have heard about that. One Health.
Okay.
It was started in 2020. Recently, it's been just only 4 or 5 years. And there are some websites also. There are some researchers and organizations. They are trying to actually bring all together. It is One Health. It is not human being. It's about the plants which are present in our nearby environment. It's even about the animals. It could be pet animal. It could be wild animal. It could be just normal some animals which are feed for us. The One Health concept is there, and slowly it is just getting the just like network pharmacology, it is getting the awareness from the people, and the databases are now they are in starting positions. Sir. There are I think so 2 3 databases I have seen, especially for this one EMR, emergence of resistance against various medical conditions. That thing is also coming because that only this One Health concept came into the picture, and you can
Mhm.
explore that One Health concept, and you'll get various databases, target databases also you can identify, sir.
Okay. And last question, sir. We have
Yes,
we want to search the already marketed drugs, want to build the network for the new disease. It is
yes,
having potential.
yes, sir.
So in that case, actually, we have to search for this like some databases for the drug with the
Yes,
drug, and we have to search the disease target only, which disease we are targeting, or it found potential of that.
yes, sir.
Is it like that only? We have to proceed further.
Yes, sir. In that, actually, you can do network pharmacology also, where I'll show you in the next 5 10 minutes. I'll show you. There
Okay.
is a database called String Database where the approved drugs or phytochemicals directly you can search there. It will give you
Okay.
network. And from the network, you can identify the reasonable protein structures. And if the
Mm-hmm.
protein is associated with the disease condition, and you can say that, yes, I am repurposing this existing approved drug molecule for a different condition. Again, you're bringing some, for example, aspirin. You see that before 1990s, nobody knows that aspirin can be given for cardiac complications. Now
Yes.
we have seen, and even
Cancer also.
during COVID-19 also, because to control that, whatever clots formation in the heart, that was
Yes.
prevented by aspirin only. It is
Yes,
part
sir.
of the regime because we
Yes,
never
sir.
expected that the platelet aggregation can be explored for aspirin this much
Yes, sir.
bigger. We don't
Yes,
know
sir.
that
Yes, sir.
part in 1990s. It is
Yes,
repurposed
sir.
compound only.
Yes, sir. Yes, sir. But the thing is that when we go for the marketed drug for the repurpose, one end one side we know that
Yes,
our non-drug is applicable to this target.
yes,
So by
yes.
using only one side information, other side of information like disease, we don't know which disease it may bind or it will bind.
Yeah,
So
that you need to do
that
systematic approach. You need to do systematic approach where the first part will be your pharmacology exploration, target identification, target
yes,
validation. After that, you're trying to do some in silico modeling and followed by in vitro and in vivo. When you have this entire things fall in
yes,
place, you can
okay.
confidently go ahead that you can propose, yes, this aspirin can be used for cancer. It's not a new
Mhm.
story.
Yes,
Aspirin can be used for cancer.
yes, sir, yes, sir.
There are clinical trials are going on, and they are in phase 3 stage right now. In
Yeah.
some conditions, aspirin can be given as the first frontline drug to control cancers.
Yes, because actually what I seen is a aspirin is basically inhibiting COX, and COX is more expressed in the tumor.
Yes, sir.
That was the philosophy they proposed actually
Yes, sir.
to be a cancer-like drug.
Yes, sir. Yes,
Thank
sir.
you very much, sir. Thank you very much.
Yes, sir. Thank you so much. Uh, is there anyone?
Yes, sir. So I have some doubts, sir.
Please, sir.
Sir, like you suggest some web servers for target prediction, and as you say, Swiss target prediction is the mostly used. Sir,
Yes.
there are two more databases,
Yes.
AdmitSAR and AdmitLab. Sir, are they trustable or not, sir?
AdmitSAR is not for target prediction. AdmitSAR is for toxicity prediction as well as it is well known for that sake only. Okay, ADME
Okay.
prediction as well as toxicity prediction. But in the latest version of AdmitSAR, that is 2.0 and 3.0 version, they
Yes,
are
sir.
giving some endpoints where the protein endpoints they are giving. But I would recommend Swiss target prediction is proper tool, and apart from that, you have one more tool called Pass Online. Pass
Okay.
Online. It
Okay.
is
Okay.
very age-old tool. It is having almost close to 20 years of existence. Pass Online, which is free, but you need an account to actually work with that. And apart from that, you have Binding DB database is also there. You can simply give the smiles notation or you draw the structure. You'll get that target's information. Binding
And
DB.
sir, what about AdmitLab, sir?
Admit Lab also will give you, ADME predictions only. I don't recommend Admit SAR and Admit Lab. It is my personal opinion, okay? But
Okay.
you
Okay,
can
sir.
try if it is validated properly, you can do that. Because
A-
all these databases are built based on the literature only.
Okay. And sir,
Okay.
using single database is fine, or we should prefer
So
more than one database?
single database is never recommended.
Okay.
You need to explore at least two, three
Yes,
and
sir.
combine them because you have a tool called Venny.
Yes, sir.
Yesterday also, sir has shown that use the Venny tool to combine all these three targets information which you obtain from three different databases, and it will give you common targets. So
Okay.
use those common targets for next exploration rather than only relying on one database.
Yes, sir.
You'll never know because in the database also they will give you a prediction score of 0. 0.6, 0. 0.7, 0. 0.8.
Yes, sir. Yes, sir.
So you cannot rely because they are only saying that yes, I am only 80% confident, not 100% confident. So you must take the chance and you need to explore the other databases also for avoiding the complications in the next stage.
Oh. And sir, as in methodology you explained, for validation, we can go for molecular docking, in vitro experiments, and in vivo experiment. So we generally do in vitro experiments to confirm is it inhibiting or activating. But as I have planned to develop a formulation,
Yeah.
so should I do in vitro experiment before formulation development, or should I continue with formulation development and then I should go for we should go for in vitro experiment?
Usually, when you have in vitro data as well
Yes,
as
sir.
in vivo data, then only you'll go for formulation. If your compound is not showing activity
Yes, sir.
alone, I am
Yes,
telling
sir.
that alone means
Yes, sir.
only API.
Okay, okay, sir.
Only the phytochemical or drug has to show first biological activity in animal model. Then
Yes,
only
sir.
you'll be looking for formulation and you'll be looking for whether the drug is reaching the system, whether the drug is reaching a particular tissue,
Yes,
and
sir.
either it is pharmacokinetics are matching or not, that you will be checking. If
Okay, okay,
your
sir.
compound is not showing the biological activity in the in vitro, in vivo,
Yes,
then
sir.
why you are playing with formulation? First pharmacology, then only
Yes, sir.
Sutex. Then
Okay,
only
okay,
the
sir.
formulation will come into the picture. Then you prove the kinetics, and
Yes,
there
sir.
is no, stopping for your compound and its formulation.
Okay, okay, sir. And sir, one more, sir. Can we get paper published just after docking and MD, or should we need some in vivo and vitro data?
Just with the network, you have papers. Right now, there are n number of papers. Yesterday
Oh,
also, Anup sir has shown the timelines. In
yes, sir.
2020 itself, I think so. There are more than 7,000 papers.
Yes, sir.
7,000 papers purely on network pharmacology without adding any MD and molecular docking.
Yes,
And the
sir.
process is like this: build a network pharmacology, perform molecular docking. You will be getting a paper worth of 3 impact factor. If you want
Okay,
to
sir.
go higher, perform the MD simulation, you'll get more than 5 impact factor.
Okay,
Perform
sir.
in vitro, you'll get definitely more than 5 impact factor, and even above, you can try. You have
Okay.
in vivo, go for nature kind of paper.
Okay, sir. And sir, one more, sir. Like, should we continue with selecting disease first, and then we should find a plant and there its phytoconstituents, or should
So
we select a plant first, then based on the targets of that plant, we should select disease? Because as in my case, what I did, took first approach, like I select a disease that we
because
see,
we have an
like,
established, LBDD and SBDD, ligand-based drug discovery, structure-based drug discovery.
yes,
In
sir.
both ways, it works actually properly in silico modeling. Similarly,
Yes,
in
sir.
the conventional drug discovery also, it will go in both ways.
Yes,
Where
sir.
you can start from the phytochemical perspective, but we
Yes,
are in a time where the time lines, the scenario which is making you to understand even from ligand perspective. Like if you have phytochemical, a
Yes,
plant,
sir. Yes, sir.
within no time, within half an hour, you will know that this phytochemical is known for what kind of activities.
Yes,
So
sir.
your question is no longer, a question mark, because within half an hour you will be solving the other problem, where
Yes,
the
sir.
disease point also you will get within no time with proper
Okay.
validation.
Okay.
So you are focusing on the disease also now. On the other hand, also you will be getting information. For
Okay.
example, your focus is on disease like COVID-19, like
Yes.
diabetes, like some hypertension. Now, within no time, you will get a list of phytochemicals. Your second question again will be solved. Your
Yes, sir.
starting point may be in any side, but within no time, within a single day, within half day, within 1 hour, you'll
Yes,
be
sir.
solving the second question. On what
Oh,
disease I need to work, or on
okay.
what phytochemical I need to work. Both questions will be solved.
Okay, sir. Thank you, sir.
Yeah, there are some questions I think so in the chat box. I'll try to answer them as well. But before that, let me ask the organizers: am I allowed to take further?
Yes, sir, you can continue, sir. Show the
Yes, sir,
demonstration.
you can continue. You can continue.
If the people are interested, I cannot actually take their valuable time, but I'll try to show what I know actually for performing few things. So I guess I need to share one more paper. This is the latest work what we have done. I'll show you quickly. It is containing almost 90 slides, but I won't take that much of time. I will try to finish this in 9 to 10 minutes.
Please share your screen, sir.
Yes, sir. Yes, sir. Okay. I haven't shared the screen. Can you see now? Can you see now?
Yes, sir. Yes, sir.
Yes, sir.
Yeah. This is about one
Yes,
paper,
it's
one work. One of my master student has worked. It's about network pharmacology and in silico evaluation of Ayurvedic formulation of an Indian company. And disclaimer, I am not representing that particular company. I just only took from the database, and which we try to explore multi-targeted management of type 2 diabetes. It was done by Prachi Katse. She's from, luckily she's from Sandip University, not Sandip University, Sandip Institute of Pharmaceutical Sciences. There is associated college, sister concerned college is there. She's from there only, the B.Pharm student. And she is my master student. And we try to take actually two formulations from one of the Indian company, that is Katakadagadagadi Kashayam and Nishakatakadi Kashayam. There are two formulations which I am unable to pronounce it properly. And these two formulations were known to treat diabetes condition, and also which is also used for diabetic neuropathy. So we tried to explore these two formulations which were produced by PS Warrior Arya Vaidyashala Kottakkal. It is simply called as Kottakkal Arya Vaidyashala. It is very famous, and across the India we have we have almost close to, I don't know how many, more than 100 outlets are there. Maybe in your town also you have one Kottakkal Vaidyashala, which is very popular one. And we took these formulations. What we have done, we have decoded, not decoded in the website itself. They mentioned clearly what plants they are using for preparing this kashayam. And there were 12 plants in one kashayam, and there are almost close to 8 plants in one kashayam. Totally, there are 20 plants are there. So what we have done, we have used a database, PubChem database. Let me show you the entire scheme of work. We have taken this anti-diabetic formulations, and we identified the helps from their own database, and also various literature also we have seen. And we have explored 2-3 databases. We started with the Dr. Duke's database, PubChem also, and we have seen even IMPACT database also for understanding the phytochemicals present in these herbs. And later we got the chemical structures, and we have drawn some chemicals which were not available in the databases. Then later we identified targets from Swiss target prediction and as well as the Stitch database. And we have used even Pass Online also for some chemical substances. Then later we have performed some drug likeness screening, especially Lipinski, and some drug likeness screening protocols are there. And we got some handful of compounds. And for them, we built network pharmacology. So network pharmacology came in between. But for building network pharmacology, you have to do all these stages. So network pharmacology is not coming out of some imaginary world. You have to do all these 4 or 5 stages to get network pharmacology. Then after that, we have done molecular docking followed by MD simulation, And finally, we propose that there are five chemicals out of more than 1300 chemical structures which are present in these two formulations. Out of 1300, we are proposing five chemical structures which are showing the best activity in silico can be explored in vitro and in vivo models. At the end of this entire almost close to 9 months of study, we are proposing five lead molecules. Basically, they are from different chemical classes. Let me show you how we have done. These are the softwares what we have used for performing this thing. So first, let me show you the screen again. So once after shortlisting all the chemical structures, we have shortlisted 297, and these 297 out of 1300 structures. Okay, we removed few based on various conditions. That criteria I am not disclosing. So we have 297 shortlisted molecules, and these 297 were classified again into alkaloids, flavonoids, and terpenoids, because these three classes were given a lot of emphasis for diabetes condition. And from there, we got 2D, 3D structures for only 239 structures. And we got almost close to 60 structures lost because their structures were not mentioned anywhere. Then we got canonical smiles from PubChem, and we explored Lipinski drug likeness screening, and we shortlisted only 171 molecules because some molecules are violating some key filtration criteria. And we got 171, and we performed toxicity criteria also. From there, we got 153 structures, and from there, targets were predicted for only 119 structures because almost close to 30 plus structures, they don't have a proper valid target reported in the literature in any database. So we built only network pharmacology for 119 structures, and we removed 30 structures there. And out of 119 structures predicted targets, we shortlisted 8 targets. And we explored these 8 targets, performed molecular docking for 119 structures, and we shortlisted only 5 compounds. And for those 5 compounds, we performed 30 different MD simulations to identify the top 5 molecules. Okay, I'll show you those lists. List of chemical substance. This is a SMILES notation. I'll quickly go through. This is the Swiss ADME prediction. How we'll be doing it. I'll show you in the demonstration if it is possible. And let me show you the toxicity and target information. And from here, I think so network pharmacology will come into the picture. And these are the targets which we found from disease perspective. We use a string database and stitch database to get the diabetes associated target proteins. And similarly, we got the targets from chemical perspectives. Also, you can see here brucin is from a chemical, is from a plant. And for that, dipeptide DPP4 is there. DPP4 is definitely a valid target in diabetes. Similarly, you have SLC, you have ATP sensitive potassium channel, and you have sulfonylurea receptor, glucagon receptor, HDAC is there, and all these are from chemical perspective. And we have totally here targets from chemical perspective, targets from even disease perspective. And with that, we build the network pharmacology. And I skipped all the stages. And here you can see that there are different layers are there. And this is, so the last layer, whatever last layer I mentioned here, this layer is about phytochemical. And the phytochemicals are connected with the internal layers of target molecules. And among the target molecules, we applied the degree filter. And in the degree filter, we kept in the center where there were 8 targets. These 8 targets we explored in the molecular docking and MD simulation. There were targets. They're also having connectivity with all different phytochemicals. But we have proper, shortlisting criteria based on earlier I mentioned. We have betweenness, closeness, nearness, and apart from that, the most important degree filter. We applied all these 4 criteria, analyzed properly, and we took this validated 8 targets for further exploration in silico modeling. And these are the 8 targets. And in this, we again dropped one more target, that is hormone sensitive lipase later onwards. Based on the existing proteins availability in the PDB database, we dropped this also. Out of 8, we performed only docking for 7 targets. You can see here PPR alpha is a validated target. GSK3 beta is a target. Since 2010, it was given more emphasis. DPP4 is well-known one, and STAT3 is well-known one. HDAC1 protein is also connected to diabetes properly, and PPR gamma is there, and alpha glucosidase is well-known. So these 7 targets were performed MD simulation, a molecular docking by using molecular virtual docker. And here I am showing you a number of slides. This is about docking data. comparison with co-crystallized ligand. And I'll show you 3-4 slides quickly. Alkaloids. Among that, I am showing only 5 top alkaloids which are better than co-crystallized ligand. You can see the Moldog score was mentioned here, which is minus 182. Basically, this is generated with the molecular virtual docker. Here, the score is always in hundreds. Unlike Autodoc and unlike Schrodinger, where in Schrodinger and Autodoc, you'll be having minus 10, minus 8, minus 9, something like that. But in this particular software, if it is more than minus 100, it is best to score. But when it comes to this particular chemical from the phyto, the background, which has got minus 182, which is much better than co-crystallized ligand. So we felt that yes, this compound is better than co-crystal. Similarly, the same alkaloids against GSK 3 beta, there is one chemical which is very higher in terms of molecular docking score with co-crystallized ligand. DPP4, we got cyclinin, which is having best score compared to co-crystal structure. PPR gamma, and similarly STAT3, similarly for alpha glucosidase, and finally HDAC1. So similarly, for flavonoids also we explored for alkaloid. So flavonoids also against the same 7 targets. We have the data like this. Terpenes, we have the data. And we have done even molecular dynamics. Anyway, you have seen that, and I am skipping for the time being this entire videos. And this data, let me show you one conclusion slide for this entire work. Yes, this is the conclusion slide for this entire work. Out of all the explored docking, 41 alkaloids, 19 flavonoids, and 49 terpenoids, and we shortlisted 5 top alkaloids, 5 top flavonoids, and 5 top terpenes. And these 15 molecules were conducted with 10 nanosecond simulation. 10 is very less time. And out of that, 1 alkaloid, 1 flavonoid, and 1 terpenoid we are taking for 100 nanoseconds. Okay, so likewise, we shortlisted these close to 5 top molecules, and these top molecules are this one: dihydro, longiquartine, rheumatin, beta carotene, and ocroliphamin and camphorol. So these molecules are actually top molecules, and these phytochemicals we are exploring into the next 100 nanosecond time scale, and after that, it can be taken for in vitro and in vivo biological activity. So this is the outcome of this particular activity. So let me show you by taking one example. First of all, I will show you the string database. So this is a string database. You can see here, once you see in the Google, if you type the string database, you will directly get like this. So in this, there are n number of options are there in the left hand side. I'll show you one important thing, that is pathways, process, and disease. For example, if you want to understand how the paracetamol, if you type paracetamol, and it will give you an entry here, and you select here Homo sapiens, or else you can select a different organism also. You can see here there are n number of organisms were mentioned here. You can scroll down, you will get close to 100 organisms. In this, virus is there, fungi is there, staphylococci, you can see that, and even the mutated organisms also there. So I am selecting Homo sapiens only because I just want to see paracetamol can be used for like how many types of target proteins. I am not talking about disease here. If you click search here, it will give you some options here, which is coming from Reactome database or gene ontology database. So it is telling there are 27 proteins which were connected. So it will give you a network here. It will give you proteins information where these proteins are having proper affinity towards your paracetamol structure. Similarly, you can try for any phytochemical. If you want me to try for curcumin, I will try for curcumin. I'm not giving any disease perspective. I'm giving directly Homo sapiens. You click here, you will get n number of pathways. You will get more than 5,000 pathways here. Can you see here? Each pathway is having close to 76. I will take this one, which is having 116 pathways. If I click continue, it will produce a network pharmacology with n number of proteins, which is even confusion for us. So from this, again, there will be analysis tools are there. You can actually explore all these analysis tools. Okay, so there will be nodes, edges, and degree filter. Everything is there, and it will give you some editable options also. And out of this, you can bring some, the valid targets by applying all enrichment analysis. You will be shortlisting few structures or few protein structures which are connected with curcumin alone. And those genes or maybe proteins can be connected with the disease by using, again, a different tool called as dysgenate, something like that. And this is again I am showing for phytochemical. For example, if you ask me a disease condition, for example, you want to see diabetes. In diabetes, again, you have a number of conditions. Diabetes mellitus I will take, and again Homo sapiens only. I'm not changing any organism. You can see that. What network? The same network I kept in my slide as well. Okay, so these all proteins are interconnected. For example, if you click on this INS, it will give you information about insulin A chain. Similarly, if I click onto this particular PDX, what is this PDX? This PDX is nothing but pancreas homeo box protein 1, which may be somewhere connected with diabetes condition. Similarly, here you can see that PPA, something protein phosphatase 1 regulating subunit 15B. This is also connected with diabetes condition. And ITPR. So these are all individual proteins, inositol triphosphate receptor type 3 protein. This also might be connected somewhere with the diabetes condition. So like this, actually you can get a number of disease and their target profile. So this is one side of your network pharmacology. That is whatever you got, your job is to analyze. Okay. The second thing, let me show you the impact database. For example, you want to understand the phytochemicals. For example, you have a plant. Acha director, for example, if I'm typing it properly. Oh, the name itself is wrong. Let me type here Acha director. Indica. Okay, can you see this Ajaradicta indica? This plant information was mentioned. If you click that, you'll get information about the phytochemicals. Can you see here the fourth column is talking about phytochemical name. It contains almost close to 418 entries. Means this plant contains almost close to 418 chemical structures. And these chemicals can be classified across a number of classes. It can be, alkaloid, glycoside, terpenoid, and tannin, something like that. Okay, for example, for one chemical substance, nimbiol, I will try to see whether it is having any. This is another database called BindingDB. So there is a tool called as find compounds for my target. Find my compounds target. Okay, it is just like this target prediction. Your job is to actually give the Smiles notation of this particular compound. Let me open this compound. It will give you Smiles information here directly. Directly copy the Smiles information and paste into this database, and it will give you structure here directly. And your job is to actually select the database with similarity of close to 0.7, 0.6, 0.75. That's up to us. If you click go, it will give you the target's information. So this nimbiol was mentioned somewhere in the literature to give some inhibition against a particular target molecule or a particular disease condition. If it is working properly, you will get the result within seconds. Network is there. Okay, internal server error. Sometimes you can actually upload the structure also. Let me upload the structure. I think so. I got one structure downloaded in the morning. Take all files. Okay, it's not there in this SDF file format. I'll try to actually search for curcumin. Let me try for curcumin. So I got the curcumin and I'll take the curcumin Smiles notation. I will paste it here. The key 0.6. No, there is something issue. The server is not available. But usually, if you are connected to personal network, it will give you. It will give you the list of targets. From there, you can actually get the Uniport ID. With the Uniport ID, you can explore the dysgenate, and which will give you the disease and gene association by using dysgenate. This is a dysgenate platform. So since last 2 months, it is now proprietary version. And if you're taking an account, and you can actually do some searches related to diseases variants and genes and chemicals also. It need not to be a phytochemical, it can be anything. Okay, this is from the target's perspective and even phytochemicals perspective. If you have everything available in your hand, actually you can use this particular software called Cytoscape, and you can load the data and you can build the networks like this. The Cytoscape tool is very useful and one of my favorite tool to build the network pharmacology. So right now, as we are exceeding the time, I think so. I'm not actually taking the liberty of showing this particular thing. Sir, I think so. I can stop here from my end. I hope that this particular cytoscape will be definitely covered by the next three speakers.
Sir, if when you built the network with using the proteins, whichever the common you found in the both the
Yes, sir.
procedures,
Yes, sir.
so that how you connected that selected targets with your appropriate chemicals? How you are knowing that whatever the targets you selected
Yes,
by
sir.
using the different four parameter, which particular chemicals they will interacting more? Actually, you shown your case study. You
Yes, sir.
selected 7 target, and
Yes,
out
sir.
of that you found some few chemicals.
Yes, sir.
So I just want to know how you selected actually these chemical connection between the protein by using these.
If you allow me 5 minutes, I can show that.
Yeah.
I may not give actually the quick introduction about this one, but let me show you. This is about how to build a network pharmacology using Cytoscape. Can you see here the Excel sheet is visible to you?
Yes.
Telling about the Triphala formulation. In the Triphala,
Yes.
there are three forms, like three phytochemicals are, that's why it is called as three phala, like three fruits. One is Embica officinale and Terminalia cebula and Terminalia bellarica. So this is the way how to actually, how you need to give Excel sheet to play with the Cytoscape software. So this is one form where you have the formulation, the botanical, and the botanical attribute was given like this. And based on the chemical, like based on the botanical name, by using PubChem database or maybe even Dr. Duke's database, you will get the chemical information. Can you see here? There are three different colors in this Excel sheet. Umbilica officinal is connected with almost close to 7-8 compounds. I mentioned here 9 compounds, almost close to 9 compounds. So this is basically the bioactive information of your plant. This is called as bioactives. Similarly, Terminalia cebula is having almost close to 10 phytochemicals, and Terminalia bellarica is having almost close to another 10 phytochemicals. So this is another layer of network I am building in Excel. And the third one I will show you by using that phytochemical. Can you see here? I used citric acid, and I actually got the gene information of a particular target. And I am sorry, here it was simply mentioned like a gene, but this gene is actually a target molecule.
Okay.
And similarly for ellagic acid, I took almost 3 targets. Camphorol, I took 4 targets, but it is not limited. Just to build a small network, I limited this. For cebullagic acid, you may get more than 100 genes. You have to include all 100 genes. Similarly, ellagic acid, you have to include all 100 or 150 genes. Camphorol, similarly here, there are phytochemicals, which we connected like this.
Sir, one question, sir, for this slide, sir.
Yes.
So let's say there are more than 100 genes for a single chemical compound, right? That single chemical compound can target more than 100 genes. Okay. So what happened? Let's say I have 10 compounds, okay, and all 10 compounds are targeting, let's say, 100 genes. So there would be 1,000 genes targeted by all these 10 compounds, right?
Yes, sir. Exactly.
Then
Yes.
how I can then how I should screen that? I should keep all the 1,000 genes in my network, or it would be too much complex to explain.
Okay, let me show this particular slide. You might have seen that slide already, but let me just show you one more time. Yes, this one I think so. Can you see here this slide?
Yeah, yes, sir.
This figure is telling you about COVID-19 and some cancer and orange.
Okay.
1 minute. Just a second. Let me put it into the slideshow mode and I will highlight.
Yeah, I can see.
This one, this particular diagram, intersection diagram.
Yes, sir.
So blue is talking about COVID-19 target. There are almost close to 6,799.
Yes, sir.
Less than your 1,000. And here cancer is having 7,700. But together,
Yes,
there
sir.
are common genes which are close to 6,200. And when it
Okay, sir.
comes to your plant, the plant is having almost more than maybe 500. Among that, 65 are matching with COVID-19, and 24 is matching with cancer. And together, there are 354 common genes are there.
I
So
got it, sir.
354 common genes. Now we are trying to play and we are trying to actually build this network. Can you see here in the bottom the network? Definitely, it will
Yes,
have
sir.
more than 350 genes here. In that, they applied the filters and finally they selected top 5 targets.
Okay,
Okay,
those
and
target you are filtering from Excel sheet, whatever you made, you are selecting.
So that Excel sheet I will be loading into the computer, loading into the Cytoscape software. They also use Cytoscape, and I will also show in the Cytoscape only. These Excel sheets we are building manually because that has to be done to actually feed the software.
Okay.
Cytoscape, the purpose is that it will read the data from whatever file you are giving and it will build a network.
Okay.
so sir, 1 minute, sir. I did that, right? I found that. Okay.
Yeah.
Now I found after this step, I got the targets for my compound and each, as you said, I did this Venn diagram and I cleared that step as well. But still, I am getting so many proteins, means so many genes for single compound, right?
Not a problem. Let it be 500. But once after building, we have some tools to play. The filtration criteria are there in the Cytoscape. We will play. Give me 2 minutes so that I will show that as well. So right now I have shown 4 Excel sheets. I think so. I stopped here where chemical structure is there and the associated target ID. We got it here. And let me show the last Excel sheet where the target and how it is connected with the disease condition. So there is a target, which this target is from chibulagic acid, but from dysgenate, this particular target is connected with a neoplasm, a cancerous condition. So this we got from dysgenate database. So now I have 4 Excel sheets. Let me load these 4 Excel sheets into the software. So can you see here, I opened the Cytoscape software and I'm going to the file and I'm importing the file from import option, and let me direct this and I am uploading the first form and it will give you an entry in the right hand side, and there is a pop-up window which will tell you that we have to give here the attributes. The formulation is always the source node and botanical is a target node. Once after you are giving this, you have to click OK and there will be an entry here and you will be seeing on the screen there is a Triphala is connected with 3 plants. Can you see this architecture? And after that, and I will be importing another one, that is botanicals and associated bioactives, where I have 3 plants, which is my source, and the chemicals, which is my target node. And after that, and I'm importing one more thing. Can you see now? The tree now became bigger, and each and terminal cebula or maybe whatever Bellarica is there, each one is having almost close to 8 and 9 different lines. And let me load one more file, which is basically the biological activity and target information. And here, the biological activity and node also like the assignment I have given, and you can see now that it has become scattered. Then let me load one more thing. That is the final one, the disease thing. And I got this, and already this was given as a source, and disease ID I will be giving as a target node. And I got this, and here it is totally clumsy, and you cannot see that it is not a proper one. Okay, so let me do one thing, and I am just merging all these things, all these 4 sheets into one particular network, and I am merging all these 4 like this. Let me select union. I am just getting, and you can see there is a clumsy network is there. Let me put this into one proper layout, that is usually circular layout we'll be using. Can you see now it has become like a proper network? So from here, for example, you have more than 500. Now I don't have 500, maybe close to around 50, 60 are there. Now your job is to actually filter this entirely. Before that filter, you have to analyze this particular thing. There is an option called analyze the network. Once after clicking the analyze network, you will get few things here. Can you see in the bottom? There is a kind of a sheet is there which is telling you about degree, betweenness, neighborhoodness, number of directed edges, radiality, few other parameters are there. And here on the right hand side, you can see that it is telling you that there are 94 nodes, 114 edges. The average number of neighborhood is 2.4. Network diameter is 10. All these are actually qualitative parameters. If this average number of neighbors, if it is more, it is actually a good, the network pharmacology model. And here, for filtration, let me enable something here. Can you see here when I click here, you'll be getting column filter, degree filter, and topological filter. I will apply just only degree filter and I'll show. And here you can see that everything is in yellow color because everything got selected. And here among this just a short, almost close to around 50 number of target genes only available here. Among this 50, we have maximum connectivity of 13. Okay, and minimum connectivity of 1. For example, if I want to filter all these things, what I will do, I will apply the minimum number of connectivities I required and the maximum number of connectivities I required. If you remember, I told you earlier that there is a gene or a protein which may have more than 50 connectivities. We have to exclude such kind of protein also because you'll be having off-target effects. So for that purpose, let me see if there is only one connectivity, it is not a valid target. What I will do, I'll try to actually apply like this. Can you see when I'm applying, the connectivity is now becoming lower. Okay. If I do like this, again, the connectivity is going lower only. If you see here, there is only 3 targets. 1, 2, 3 are there. This target is there. Only 3 targets were there between 6 to 9. So this, whatever filtration, degree filtration criteria is up to us. We have to take that decision by understanding that particular protein target. If you click here, that protein target information you will get. If you double click here, somewhere you will get that information in this software itself about that particular target. I'm not getting here, maybe I need to load some more things to show you that information, the nodes information. Okay, so these 3 targets may be a valid target for my Triphala combination. Okay, so this is a way how you can actually manually build the network pharmacology from the herbal formulation perspective. Triphala is a herbal formulation, and here again you can move all these things and you can properly keep them into center, and there is a lot of style options are there. You can change the colors, and you can make it more beautiful. So many things are there to explore in this cytoscape that itself will take almost close to 2 hours of session to understand the architecture of cytoscape.
Actually, this also can be built. Network can also be export from the database, Stripe or Stitch, whatever you.
Yes, sir. Exactly. From the string directly, you can download that disease-associated network pharmacology, or maybe phytochemical or drug-associated one, and you can apply here. You can
And the same thing it is there.
analysis.
But
You can
all
perform
the
all
four-
the analysis here.
Okay, so this Stitch or database, you already shown this closeness, betweenness.
Yes,
So
sir.
this cytoscape, does giving the same values or it will calculate in the different way?
Same, sir. Same. Same, sir.
Okay.
And you have actually the apps are there. Can you see here the string?
Yes,
Directly,
yes.
I can load from the string here.
Okay.
There are almost close to 10 apps are there where you can
Mhm.
perform every analysis in this tool itself.
So whatever you present, this is applicable for formulations, and Stitch
No,
is
no, no. Single chemical also you can do. Single paracetamol, single routine, or any flavonoid, any alkaloid you can do. Or
Okay.
disease also directly you can do.
Okay, but only one set of, we have to search the literature for one aspect, and other we have to search the databases, and
Yes,
we
sir,
have to merge both the targets.
Exactly. Yes, sir. Yes, sir.
Thank you very much, sir. Very nicely you presented, sir. Thank you very much.
It's better, I think. So I need
Actually,
to
I am
stop.
from your sister concern only. I am from SVKM Dhule actually.
Oh, nice, sir. Nice to know you.
I seen actually your this all those things. So thank you very much, sir. You nicely presented. Thank you, sir. Thank you, sir.
Sir, Gandhi sir, I'm very sorry and extremely sorry for extending this session almost for 47 minutes. And I'm very much, happy that you people are, interacting. And it's not a kind of boring session, I guess.
No problem, sir. It's a wonderful
Sir, over
session.
to you, sir. Nagaraj, sir. Please go ahead,
Yeah.
sir.
Yes. Gandhi, sir. Dear participant, I think if you have any questions, you can ask.
I guess there are something in chat. Can I take them directly?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Uh,
Of,
Rashek. Yes, sir.
yes, sir. Gandhi, sir.
Yes, sir. Yes, sir. Please.
Okay, sir. Sir, can we use network pharmacology
So
for
please,
design?
sir, there are few things on the chat box.
Yes, sir.
We can address them.
I'm reading them. So we can use network pharmacology for designed molecules to predict target. I missed the initial part. Yes, it can be done for anything. You can actually predict for design structures also. And ligand-based drug design is good. Sir, if my focus on multifactorial disease means it requires poly drugs as target molecules, but it may show number of side effects, then how to tackle these side effects? Yes, for that purpose only, we need to have proper understanding about the disease pathophysiology, the pathogenesis, and what proteins are connected. Based on that, you can solve that off-target effects or side effects thing. And somebody answered hydrogen bond, ionic and pi pi, good one. And the sandwich interactions is rotatable of atoms, like RCSP, any other PDB generators are there. As of now, I think so. PDB is the best one, and there are even some databases are there. Binding DB also will provide you the protein structures. You can download from there also. And another one asked, Swiss target prediction is not working these days. It will work, but only thing is that you need to connect to a different network. I can say that in my own network, the Swiss target prediction is not working. So you have to connect to your mobile network. Definitely it will give. But what happens? These are publicly funded databases. Usually, sometimes there will be downtime of the server. You need to try after 4 or 5 hours, maybe sometimes even after a day. So definitely, Swiss target prediction is working. Today morning also, I have seen that it is working. So can we go for network pharmacology for any synthetic drugs? Yes, absolutely, you can follow that. Sir, can you please share your email ID? Okay, I will share that, no problem. Sir, for any cytoscape, cytoscape is free. You can download it. You can install it in your computer. It doesn't matter whether it is, the computer is having high-end computational power or not. You can install that. So TB database, yes, sir. I will try to share that TB database. Anyway, I have that in my computer. Sir, over to you, sir. I think so. Questions finished.
Thank you, sir. Thank you so much for your valuable time. You've already spent more than 1 hour on the question and answer session, addressing all the queries from the participants. Thank you, sir. Thank you so much. You started your presentation with the traditional drug discovery aspects, and then you moved to the challenges faced from traditional perspective, tuned slowly all the participants into network pharmacology, then the case with Narinjanin, and then machine learning, real-time examples. Your presentation is very interesting, and it has attracted all our participants. From our bottom of our hearts, we would like to thank for sparing your valuable time with us, sir. And you have shown the hands-on training on different softwares. Thank you so much for that. As a token of appreciation, we would like to accept the certificate of appreciation from our university, sir. Thank you so much, sir, for your valuable time.
Certificate not showing once you shared it.
Yeah, I'm waiting for the certificate to see.
Gandhi sir.
Still, it is getting shared.
Yeah, yeah. Gandhi sir, are you there?
Maybe there is some issue. It's not a problem. Yeah, I
Wait,
guess
wait,
I
wait.
shared my email ID in the chat box. You can see that. If anybody want collaboration, you can approach us. We are very happy to associate with academic people or industry people.
Wait,
Thank
sir,
you
1
so much
minute.
for patient listening, and I just want to thank Mohan Gandhi sir and Nagaraju sir for giving me chance to speak to you guys. And I just want to thank Dean sir and Associate Dean sir and the chairman of Sandip institutions. Thank you so much for providing this opportunity. I hope, I guess,
Yes, Rajshekhar sir, there is a technical issue with Gandhi sir.
no
Yeah.
issue.
Yeah, we will share the certificate to you.
No
Yeah.
problem.
Yeah. Thank you, sir. Thank you for your wonderful talk. With a short time, you are accepted my invitation and give the wonderful presentation on the network pharmacology. So we have to collaborate in the future endeavor also, sir. Definitely. Yes.
Sure.
Yeah.
Sure.
Thank you, my dear participants. You listen very patiently last 2 hours, and I hope you get some good knowledge on this network pharmacology. So if you want any collaboration, already sir is shared the mail ID. You can directly contact with the sir. Otherwise, you can contact me also. So we both are very good friends and colleagues previously also. Okay. So I will help you in that way. So we will meet you tomorrow in the same time at 10:30. Okay. So thank you for all. Thank you, sir.
Thank you, sir. Thank you.